Well, thank you, Claretta. You pretty much made all the major points I wanted to, which is great, because now I can just fill in the details and give you some images to make it a little more vivid. This is a really, really profound moment and very difficult questions as we've seen. I spoke at a conference at Harvard last spring, and I thought I'd start by showing you this interesting advertisement, a poster they used, editorial aspirations. And what's so fascinating is the hands, reworking the hands. Aristotle called the hands the tool of tools. And what an amazing thing that it's our hands that are now actually editing ourselves, potentially. This is actually not something conceptually new, just much more powerful and profound. I guess you can read this. Maybe I should read it. That means it's a talk, right? Mm-hmm. Nor can any person, whatever kind of religious phrases he may use, fail to believe that if nature and man are both the works of a being of perfect goodness, that being intended nature as a scheme to be amended, not imitated by man. As a physician, I've spent much of my life thinking about this very question. What is the proper boundary of what we do, especially as our medicine changes and becomes more powerful at basic physiological levels? The imperative of healing is perhaps the most profound and uncontroversial intervention in nature that one can think of. And it's received the full endorsement of the human family. Yet what is a disease? And what does it mean to have the power to go to the very foundations of our nature? We're the culmination of a trajectory of increasing comprehension and control within the universe, with the point of an evolutionary process that has put us actually full circle now to return to the very foundations of life itself. What Hans Jonas, speaking more broadly, is called the progressive scale of freedom and peril. 70 years ago, Aldous Huxley, anticipating the transformation of human life through advances in biology as the final and most searching revolution, asserted this really revolutionary revolution is to be achieved not in the external world, but in the souls and flesh of human beings. In the decades since the first publication of Brave New World, amid the accelerating pace of discoveries in genetics, developmental biology, and the laboratory production of life, there's been an increasing appreciation of Huxley's prescient concerns. Yet throughout this period, limitations in our tools and technologies for specific and efficient modifications to genomes have been a major constraining factor for advances in biotechnology. Now, however, in what MIT Tech Review has called the biggest biotech discovery of the century, CRISPR-Cas9 and related techniques promise easy to do, inexpensive, and highly precise genetic deletions, insertions, and functional manipulations of genomic processes across the full spectrum of living beings. This holds great promise for advances in agriculture, animal studies, and basic biomedical research and therapy, but opens fundamental questions about our role within the natural order and the use of these technologies in shaping the human future. CRISPR-Cas9, which is adapted from an immune system deployed by bacteria to protect themselves against virus, has been, well, it's an acronym, you see, you get it, that's what the acronym spells out, CRISPR. It's a naturally occurring agency in bacteria to dice up, splice, cut viruses. It's been described as a genetic scalpel or molecular scissors, but it's more rightly compared to a Swiss Army knife. It's composed of two basic elements. There's guide RNA that identifies, and it's a sequence of RNA that identifies and attaches to a specific stretch of DNA. And then there's an endonuclease, that's an enzyme that cuts nucleic acids, which allows a range of expression of the combination of these allows a range of operations on the gene segment. So you got the guide RNA and the cutting apparatus, but the cutting apparatus can be replaced with a bunch of other substitutions that can do other things than cut. As scientists continue to explore the CRISPR system, which has many natural variations, and as they selectively engineer clever new modifications in its targeting and operations, they're developing a whole new genetic toolkit with functional applications at every level of genomic process. So that's a very important thing to grasp, how cheap, quick, and easy it is to do. Jennifer Doudna, who was mentioned a minute ago, says advances in gene editing are enabling us to rewrite the very language of life. We're standing on the cusp of a new era, one in which we will have primary authority over life's genetic makeup and all its vibrant and varied outputs. I'm not sure I agree with the primary authority, but if authority means the ability to author something, we can author new variations. Just to quickly go through some of these, there already are nutritious new fruit crops, not done with CRISPR, but earlier forms of gene editing. But now we're getting very close to new crops with CRISPR. New forms of photosynthesis that are more efficient are being worked on. Complex action on multiple genomic elements, editing insects like Zika to try to remove it from the environment or else make it unsusceptible to the mosquito, unsusceptible to the virus. There are now new forms of CRISPR that can address themselves directly to RNA. It's just a whole slew of amazing new possibilities for identifying new drug sources, doing gene editing in up and running patients, and setting up for transplantation of organs by eliminating retroviruses. Most importantly, and more significantly, however, is the intervention into development and the flexibility in studying development. We've gone through a century of advances in molecular biology. Now in the 21st century, the era of developmental biology is upon us, and we want to know how those molecular components knit the organism together to produce living beings. And that brings us to the very foundations of developmental process and their genetic mechanisms. Very fascinating moment. My colleague at Stanford, Stanley Key, a very good and responsible scientist, says we should think carefully about how we're going to use that power. Amazing possibilities, wonderful possibilities, and a lot of very disturbing suggestions that are coming out already. With all good intention, Kevin Esvelt, who's an authority on gene drives at Harvard, a very nice person to talk with on his web page, he says we're coming to the point where we can correct a major flaw in evolution, namely that a very major flaw in nature, namely that evolutionary process has no moral compass. So he's in favor of retooling entire ecosystems to function more smoothly. Others suggest that we can do a range of experiments, and of course there's already gene transfers that have made phosphorescent mice, but you can imagine with CRISPR the amazing possibilities. I've suggested in my classes for years that sooner or later we'd have a new kind of Dr. Seuss zoo with creatures of previously unseen character. And of course there's the possibility of creating human-animal chimeras that are strangely troubling. I'm not against all human-animal chimeric creations, but I think we have to be very careful what we do in that regard, not to violate human dignity. Others have suggested, including George Church, that we might one day reconstitute our ancestral species, or our cousins like Neanderthal and Denisovans. That there's some reason to think that we can do that. We've now got basically the entire Neanderthal genome. And of course, you know, George Church wants to bring the woolly mammoth back. Like it in MIT suggests we might be able to intervene to produce the perfect astronaut. Space travel is looking more and more dangerous, but we could conceivably do it if we could do genetic alteration on a species. I doubt it, but anyway, there it is. Others, this is more fun, they suggest we just downsize the species, make us smaller so we don't use so much natural resource and we can occupy more space. Maybe the whole population of the world could fit into Chicago. But on a more serious note, there are large groups of people, as you all know, who want to retool our species to make us more adapted here on the Earth. And their projects, Francis Fukuyama was on the council with Gil and me and wrote this thoughtful book. So among the various goals for the use of CRISPR are to retool our genes so that we will live forever. There again, I think that goal is a little bit like trying to invent a pole vault that will send you over the moon. But be that as it may, there's significant interest in these ideas, and it raises a very serious challenge. I think I've had members of the Transhumanist Society in my classes at Stanford. In fact, my TA a couple of years ago was the founder. And they're intelligent, serious-minded students. We need to take them seriously. They argue that our advancing technologies offer us the opportunity to escape the constraints and cruelties of an amoral evolutionary process and to lift humanity to its next level of personal and social flourishing as genetically enhanced human-machine hybrid post-humans. So we're up against a great challenge. The powers of this technology are in a way just in step with our previous scientific advances. We've made great advances in basic physics, in synthetic chemistry, and now we're moving on to biology. In the few years since its introduction, CRISPR-Cas9 has been employed in labs all over the world, generated thousands and thousands of scientific publications, and has attracted well over $1 billion in venture capital from private companies exploring its medical and commercial applications. I think that the discussion of this is amazing. As I've gone around and talked with people, I was in Washington last week talking with some, and I'm amazed how few people have this on their radar. I guess we got a lot of things to talk about, but I'm pretty worried about it. The transhumanists are gaining power in the universities. It's a new fashion, a new wave, a new cult. And I'm much more worried that instead of leading us on to the golden age of genetically improved human beings, we're more likely to award ourselves the Darwin Award. So that lays it all out. I mean, you all know what I've said so far, but it just puts a frame around it. There are three basic issues that I think as Christians we need to take very seriously. The whole question involved in human genome editing, which indeed, as Claretta rightly said, go way beyond just questions of safety and individual private rights. They raise basic questions about what we think medical interventions are for, and also what we might do with our own species. Biomedical research raises profound questions about the kinds of research we should allow, both on animals and human embryos, and up and running human-born human beings. Overarching it all are, and I think this is in one sense where our Christian faith makes the most profound difference, and that is the vision of nature that guides our work. I don't share the visions of the transhumanists. I don't think any of you do either. The question of where did nature come from, what is human life for, and what's our prerogative in regard to our role as co-creators within the natural order. So this is Jennifer Doudna, who we've mentioned a couple times. Jennifer and I she's the scientist along with Manuel Charpentier from France who discovered CRISPR-Cas9. And Jennifer and I have been working together for the past 2 years on a project to advance discussion, deliberation, and education on CRISPR. And she's a really wonderful person to work with and has serious concerns. They don't always overlap with mine, but mostly they do. And I feel as though we're very lucky to have a scientist with such thoughtful consideration of what her discovery will lead to. So as you see, she says we may be nearing the beginning of the end of genetic diseases. And on the somatic cell level, that's a very encouraging statement, because we have well over 6,000 single gene genetic diseases that afflict our species, 95% of them with no treatment at all, no cure and no treatment. So this is blood from one we can treat a little bit, and that's sickle cell anemia. You can see the sickle cells mixed in with the regular red blood cells. And with a disease like this, it's going to be the low-hanging fruit for this kind of intervention, because what you can do is draw the blood out with a syringe, and it will include, you can mobilize hematopoietic stem cell precursors into the bloodstream, and once you draw the blood out, there are thousands of these precursors that you can then alter in the dish with genetic engineering and then put them back in. And the hope is that they will colonize, and it seems to be true, that they will colonize the bone marrow and then start putting out better red blood cells, enough to swamp out the negative effects of the sickling cells. And you don't actually have to do very much. If you get a 20–30% increase of normal cells, the patient will have no symptoms virtually. And so that's a very encouraging, and I think it'll be the very first realistic use of somatic cell gene therapy. Well, there's a few that are happening now with zinc fingers and earlier technology, but sickle cell is going to be a big deal. And my colleague at Stanford, Matt Porteous, who served on the National Academy, is pioneering in this, and it's exciting to talk with him. By the way, I took care of a patient when I was a med student at Stanford. Little boy, little black kid, he was 11 years old, and he'd been in the hospital over 200 times. So very serious stuff. There are many such diseases. This is called Miller's syndrome. Most genetic diseases are syndromes. If those of you not trained in science and medicine, genes don't affect just one thing, they affect many. They're like colors on an artist's palette that get mixed with other colors, and so genes usually express it as multi-phenotypic disorders, and they're very tragic. Among the most tragic one that afflicts children is Lesch-Nyhan's disease, where there's a metabolic deficiency. Children actually chew their own fingers off, and sometimes their own lips. So would we intervene in these diseases at the somatic level? I think that doesn't raise any major controversies for me beyond safety and maybe a little on resources, but those are pretty clear in keeping with the traditions of medicine. But then we come to the more difficult issues that Clara raised. What is a disease anyway? Is this a disease? There were 2 albino patients. They were people patients. I think of them as patients because I'm a doctor, but they were sitting in the bus the other day when I was at the Chicago airport coming here. And I was watching them. They were hanging out with each other. I guess they accept each other better than others would, but they were functioning and up fine. They just have to stay out of the sunlight. But then it's obvious that certain people consider certain forms of their body to be disordered in a social way. Pat Williams, who's a professor at Columbia Law School, was part of our ethics workshop on the CRISPR project with Jennifer Doudna, and she told us about a student she had who, there's two Chinese students, American Chinese students, wanted to have a baby, and they decided they would use a Caucasian sperm donor instead of the father because they wanted to have the baby look more acceptably beautiful to mainstream beauty standards. So I think it's possible. Some people have suggested, Pat Williams also suggested, that people want some African Americans want lighter skin and would do the same thing, genetically engineer themselves to have lighter skin. Others maybe would affect genes that put the epicanthal fold on the Asian eye. That's primarily what they're after in terms of surgical procedures. So you can imagine a great many subtle personal preferences expressing themselves, and then it can get much deeper with aspirations toward major issues of social advantage, including intelligence, longevity, basic personality styles. Fortunately, most of those things are extremely polygenic and will not be easy to intervene with without it causing many alterations to systems you didn't necessarily want to change. So I'm fairly optimistic that this will not be used in a very major destructive way, but what is acceptable? So this is George Church, also part of our working group. I've gotten to know him quite well. Had a long dinner with him at Harvard a couple weeks ago. He's a very interesting guy. To talk with him is sort of like listening to popcorn, watching popcorn pop. Don't you think, Kevin? He does everything, he just shoots out ideas, and I'm really glad he's part of our species. I'm also glad that he's not in charge of the whole thing, because I think I really like him, and I think he's really a great wellspring of a lot of creativity. And he actually has some concerns for the ethical issues. It's just that he's got a limited realm of intense laser beam focus of thoughtfulness, although he's very broad as a scientist. But anyway, he's a very eager and willing participant in these ethical discussions, and a fascinating person to talk with. But he raises some very, very serious questions. I'm going to raise a few of them in my talk. One of them is, he says that there are a whole list, a couple dozen by now, protective variants with, these are gene variations, alleles as scientists call them, protective variants with large impact, which exist naturally in our species, but only in a few people. So here are some of them. Lean muscles, strong bones. I'm not sure what the low odor thing is about, but anyway, whatever it is. More resistance to viruses. CCR5, by the way, is a direct intervention against HIV, and may very well end up being the first target of germline interventions, because if you have a parent who wants to have their lineage continued and he has HIV, he or she has HIV, you could decrease the probability of a problem with a child with CCR5 allele. So there are several more that are very interesting, and probably a lot of people would like the low risk of Alzheimer's disease gene. And of course, diabetes is a major, major problem, both for personal experience as a living person and a huge cost to our society. So really, you could make a pretty compelling social, personal, economic argument for even some germline engineering, maybe. But Stuart Newman says, I think thoughtfully, the genetic design of future offspring, even with the limited objectives of making these future children more normal, will open the door to attempts to pick and choose other characteristics, because definitions of normality will vary, as will access to technology and willingness to take risks with future lives. And that last phrase, take risks with future lives, is very relevant to our consideration here. So we're at an amazing moment. Visions of the human future, high schemes, but what does it mean? And what should we think about it? Gil and I spent a long time working on the volume that you mentioned, Beyond Therapy, Biotechnology, and Pursuit of Happiness. And it's a very difficult subject, actually. I as a physician, keep coming back to Galen. I said the physician is only nature's assistant. But what does that mean now in the age of biotechnology? Are we supposed to assist nature in rectifying basic flaws, since it has no moral compass after all? So you think about it. When we did that volume, we didn't address exotic sort of enhancements, but just what we thought the average person would more or less think was good for themselves and their children. Of course, all of us want healthy bodies. Most of us want good lives, but it's very obvious that there's something going on when in our current age with the lifestyle drugs and so forth. As Galen points out, the traditional role of medicine was to cure disease and alleviate suffering, to restore and sustain the patient to a natural level of functioning and well-being. By this perspective, the medical arts are in the service of a wider reverence and respect for the created order of the world. But now, with the powers of our advancing biotechnology, there's a new paradigm, one of liberation, technological transformation in the quest for happiness and human perfection, grounded in the widespread practice of cosmetic surgery, which has been a huge surge in that since I've been in training and practice of medicine, experience of medical realities. Grounded in the paradigm, the conceptual notion of cosmetic surgery, slowly but steadily the scope and purpose of medicine are being extended along the gradient of our appetites and ambitions to encompass definitions of life not previously considered matters of health, but of human variation or limitation. From Rogaine for baldness, you know this line, the ad says, if you're losing your hair, you no longer have a reason to lose hope. Wow. So Rogaine, growth hormone for shortness, birth control pills, Viagra, more recently Provigil, a drug that prolongs period of wakefulness up to 60 hours, good for studying, I guess. You forget everything you learned. But, and seasonale, a technological bypass to the monthly periodicity of the natural menstrual cycle. In all these ways, we've altered and revised the given frame of nature. Increasingly, we've come to expect from medicine not just freedom from disease, but freedom from distress, struggle, and even the constraints of a natural life process, from all that's unattractive, imperfect, or just inconvenient. And beyond that, we want our children, especially, to have the best possible lives. So where will this go? Will we actually find ways to intervene for IQs, better general brain functions? It's no longer sci-fi. That's the amazing thing. It's not theoretical in the same way it was before. It may have technical barriers, but now the interventions themselves are coming into possibility. A whole kind of new realm. I just put this in here because I find it kind of chilling. It's so sterile and futuristic. So recognizing all this, Jennifer Doudna convened a group of scientists and said, collectively, they said, we think these are serious matters. This led to the Gene Summit 2 years ago in Washington, and out of that came some promulgations that were mentioned earlier. Those were based on the notion, as Paul Berg and David Baltimore said, to address complex diseases like cancer and developmental disorders and so forth. We must carry our investigations to the most fundamental elements of living systems, which of course means human embryos and the embryonic developmental processes. In the volume that they produced, they made virtually no mention of embryos. And I asked two members of the group, 3 members, why they didn't do it, and they all said, well, we just felt like that's an issue that's been decided. And that struck me as a strange response, because as I'll show you in a minute, I think even if it were decided with regard to the use of leftover IVF embryos, it certainly hasn't been decided for many other uses of human embryos, and I want to get into that in a second. But just to take you back to the fundamental problem that convened the summit was germline engineering, and just as the summit was getting ready to make its final organizational efforts, Chinese scientists genetically modified human embryos. I had lunch and spent 4 hours last Wednesday with the guy who did this, and he's a very nice guy and very interesting person to talk with, and he just basically says, well, we just have a different view of the human embryo than the pro-life faction in the United States. But this goes way beyond the use of embryos. In that study, they used embryos that had a genetic problem that would not allow them to develop further. But those of us who were involved in this discussion over embryonic stem cells know how difficult it was and how many ramifications there are. This was the cover of my med school alumni magazine, which was pretty, I think, unfair, actually. And of course, you know that these concerns are encoded in the Dickey-Wicker Amendment. But now the question is, what do the definitions of Dickey-Wicker actually say, and what will they hold up? This was a very difficult discussion at the time because of the two positive human goods that were being debated. I thought this was very interesting from political advocacy, pro-stem cell, embryonic stem cell thing. We fight because lives can't wait, but they were using lives. That's what's ironic in this statement. They were using human embryonic lives. I just want to take an extra minute and play this for you, because if I can make it work without, yeah.
On my way home?
Okay, hold on, I'm going to start this over again. Okay, just to remind us all how difficult this can be.
Next summer, I'm going on a camping trip with my friends. On my way home, I'll be in a car accident and I'll be paralyzed for the rest of my life.
In 20 years, I'll have Alzheimer's. I won't recognize my husband or my kids.
This week, my mommy is here to find out that I have diabetes.
This is my congressman.
Congressman Don Sherwood.
He voted against federal funding for stem cell research.
Is he a doctor?
Is he a scientist?
Why did Congressman Sherwood bet my life that he knows best?
Help me.
Help me.
Who knows? Maybe I'm your mother.
Maybe I'm your grandson.
Maybe I'm a little girl.
How do you know I'm not you?
Stem cell research could save lives. Maybe yours or your family's. Someone you love. Only Congressman Sherwood said no.
How come he thinks he gets to decide who lives and who dies? Who is he?
Majority action is responsible for the content of this evidence.
So I'm not sure what happened to poor old Congressman Sherwood, but I thought it was kind of a, well, unfair. I mean, it's amazingly, I think it's an amazingly violent way to approach the subject. And that gets us to the most core question involved here. When does human life begin? And from a Christian perspective, I feel it begins when the human organism starts with conception. And second, what is the role of suffering with regard to human life and the role of human beings in intervening into natural processes to address the question of suffering? And I think that those are the most profound questions that we have to deal with. I want to lay out for you the scope of those questions very quickly, and then we'll have some discussion. But just going back to the fundamental question, when does life begin? That's a 8-cell embryo on the sharp tip of a pin. I believe that you can make a very strong case for the continuity of human existence, that all human existence is a developmental process, that no single stage can be separated from any other with regard to human dignity, and that we continue to develop, that the early embryonic development is just the first phase of that. So we need to take that issue very seriously, because this technology, its first and greatest scientific application from the standpoint of pure knowledge, is going to be knock-in genes, studying the development of human beings, seeing what each gene does. And that could take you right up into the realm of fetal life and beyond. So we need to bear in mind this saying, a Chinese saying, those who choose the beginnings of a road also choose its destination. So we need to ask ourselves 4 questions. Will we now endorse the use of human embryos for a wide range of studies of infertility and early development, even beyond embryonic stem cells? The answer to that, from the scientific perspective, is you could learn a great deal. And definitely they will want to do that. Right now, Dickie Wicker still holds in our country. Second, will we allow the creation of embryos, not just the use of existing throwaway IVF embryos? Will we allow the creation of embryos specifically for research purposes? So this is Rudy Yanish, who I know very well. He did the mouse studies on altered nuclear transfer that I was working on. You all know that. Rudy made the first transgenic mouse, and he 30 years ago, he told me that it took 2 years, $200,000, and he could make one gene alteration. He told me recently that he can now do the same project for $2,000 in 3 weeks and alter 6 to 8 genes. So you see what a dramatic transformation this technology is. So the third question is, and of course, we learned most of what we know about developmental biology by doing these knockout studies in mice. Now will we want a refined version of that with human beings, human development? How many embryos is it okay to use? One Japanese scientist made 581 cloned copies. Well, you'd want a standardized platform so you could alter one and not alter the other and know exactly what the genome was doing differently with the altered gene. So, and the cloning of the macaques, announced 3 weeks ago, is a signal of where that's going to go. And I think we can probably, if they work at it, scientists will be able to implant human embryos as well, which raises profound questions. But right now, the limitation of oocytes is the limiting factor. And Davos Salter, a famous quote in Nature magazine 10 years ago, said that someday they'd be made through ES cells or iPSCs. And he said, once we do this, I have no idea what kind of moral value or rights we would give to these embryos. We'll probably go through the same agonizing we did with IVF. It could be terrible to begin with, but then it'll become a fact of life. Maybe 20 to 30 years from now, we'll read in newspapers that someone made 20,000 embryos and studied their development, and we'll decide it's okay. I think we've now made sperm and egg in mice, and it's just a matter of time, I think, before we do that with human gametes. So, fourth and final question, will we allow research on embryos beyond 14 days? And if so, for how long? And according to what principles of moral evaluation and logic? And I would add a corollary to that, will we allow implantation of them for scientific study purposes? As regard to the time, the issue, of course, was long ago set in principle in the UK for the 14-day rule, and that's Kathy Nikon at the Francis Crick Institute, who recently published a paper on OCT4, the pluripotency gene, studying human development in human embryos for a crucial pluripotent marker. So you know where this is going to go, and it could lead to many, many thousands of experiments. But how long would you carry them out? Because you'd want to see, I mean, if it was just a scientific matter, you'd want to see where where the transformations occurred during development, when some genes aren't even turned on until later in development. So there's no end to that. It raises very terrible questions, but would it be moral to go beyond it? Well, the UK is now debating this. There was an article in Nature Communications about 2 months ago with that debate going on. The Hurlbut in that, by the way, if you read that article, is not me, it's my son who was interviewed on this subject. Okay, but my colleague at Stanford, Hank Greeley, said, when asked about why they said 14 days is the limit for the California rules, he said 14 days would not always be the limit. That could be changed in the future based on understandings would likely come from neuroscience. Neuroscience, what's he talking about? And of course, Julian Savulescu says it's morally required that we employ cloning to produce embryos or fetuses for the sake of providing cells, tissues, and even organs for therapy followed by abortion of the embryo or fetus. And I debated him in New York City some years ago and asked him if he'd go to, how late he'd go, 3 months? He said he'd go 6 months, and he wouldn't say after that. But meanwhile, there is, and you may think that's unrealistic, but there is a biotech company in the Bay Area that's working on harvesting fetal organs from abortions and trying to figure out how we could use them for transplantation. This is from their website. So this is not just, speculative thought. This is amazing statement from Erwin Chargaff quite a few decades ago. Science is now the craft of the manipulation, substitution, and deflection of the forces of nature. What I see coming is a gigantic slaughterhouse and Auschwitz in which valuable enzymes, hormones, and so on will be extracted instead of gold teeth. Pretty chilling stuff. Would anybody do this? What is Hank really talking about? Advances in neuroscience. This is quotes from Ernle Young, who started the Stanford Bioethics Center. I took him to the medical center the first day he arrived. I knew him very well. I disagreed with him profoundly on this question. Thus, the blastocyst or embryo is a human organism, but not a person. It does not even become a potential person until the emergence of a rudimentary central nervous system, something which is thought to happen at about 26 weeks of gestation. That's factually wrong. 26 weeks of gestation, that's you have babies in the infant intensive care nursery that are already a month old. But beyond that, and the nervous system is well established, and there's concerns that those little ones can feel pain, and if you poke them in the infant intensive care nursery, in the NICU, you'll see they move in response to pain. Therefore, as Glannon puts it, since embryos do not have the requisite biological structure to generate and sustain the mental capacities, embryos cannot be benefited or harmed. Well, if he's talking 26 weeks, it's usually called a fetus after 8 weeks, but I think they use that word loosely. So if he's talking about 26 weeks, that's a neurologic marker of sorts, but it raises terribly difficult problems. Now back to George Church, he's pointed out that with new technologies, we can actually create cerebral organoids, portions of the brain that we just don't know whether it has any neurologic functions, sensory input, consciousness, and I doubt it does, but he's raising important questions. Moreover, just by combining different factors from embryonic stem cells, you may be able to generate something that's at least primordially like a human embryo. So these are really profound questions. He's calling these human embryo, synthetic human embryo-like entities, sheaths. So very, very challenging questions. What is madness? To have erroneous perceptions and to reason correctly from them. We need to be very attentive to the human evil that is just wrapped through and through our species. I acknowledge that it's in all of you and maybe me too. Human evil is so strange. It's so so inobvious sometimes because it's clouded and covered with apparent good motives. But Francis Crick, using a certain kind of logic, said no newborn should be declared human till it's passed certain tests. And James Watson is reported to have said, and this is a paraphrase because it wasn't a quote, but that if we do these genetic engineering studies and the doctors at the birth see that the children don't look very good, that we made mistakes, they should have the right to terminate them at birth. So of course our law in this country would not allow that. If you're born alive, you're born. So I just want to close with a comment from Leo Alexander, who was a physician who was at the Nuremberg trials and wrote the Nuremberg Code. And it's a really profound thing if we get this wrong, and especially the embryo question or fundamental assumptions about what's life's purpose. If we get it wrong at the very tiny level, it may play itself out in dramatic ways like an expanding wedge. Whatever proportions these crimes, referring to the Nazi crimes that he heard about at the Nuremberg trial, whatever proportions these crimes finally assumed, it became evident to all who investigated them that they had started from small beginnings. The beginnings at first were merely a subtle shift in emphasis in the basic attitude of the physicians. It started with the acceptance of the attitude basic in the euthanasia movement that there is such a thing as life not worthy to be lived. We all know where that went. We all know the tragedy of it. Maybe it won't be so obvious in the future, but one little step at a time, one concern at a time, the powers in our hands are leading us to a new relationship with the natural world. And for me as a Christian, there is a fallen world, there is need for human intervention, but the means and the ends need to comport. That in all things, we should operate only in a way where we have a comprehensive sense of what life's purposes are, and we never violate it in the steps we take towards certain ends. That our research, our types of interventions, should always be grounded in our vision of nature, namely that it's a created order that the Lord himself entered into with the right solution to the problem, which is love. And as C.S. Lewis said, we should answer all of our problems with more love, not less love. We're both spectators and actors in the great drama of existence. Our hands are first, before they are hands, are touched by the hand of God. Our hearts and minds must be aligned with God's purposes, and only then will our actions and our lives comport with the full meaning of the cosmos. Thank you.